Good morning, students. I am Dr. Abhishek, teacher, Department of Physiology, and I am teaching you Central Nervous System. Today is my third class in Central Nervous System, and today we are going to talk about synaptic transmission, regarding which we have already started in the last class. We talked about the classification of synapse, what are the different kinds of synapse based upon the anatomical ways, then based on the nature of transmission, and based on the number of neurons involved, like one-to-one, many-to-one, or one-to-many, which is a rare type of synapse. Today we are going to talk about the mechanism of synaptic transmission. So in that mechanism of synaptic transmission, first we need to know the functional anatomy of synapse. What is synapse made of? And then we go in the physiological part of it, that how the transmission occurs. So here you can see in the diagram shown, where you can see a synapse, and this is just a broad idea for you to see how the synapse is formed. So how is this synapse formed and what is the anatomy of this? So it consists of a presynaptic neuron, it consists of a postsynaptic neuron. So there is presynaptic neuron which is from which the information passes through, that is called as presynaptic, and then we have a postsynaptic neuron. The neuron which receives the information is called as a postsynaptic neuron. Then what is presynaptic membrane and a postsynaptic membrane? I will change the diagram to make you understand what I am talking about. Yeah, you can see here, this is a very good diagram which is showing all the structures which I am talking about right now. So we have a presynaptic membrane, and then we have a postsynaptic membrane. So what is a presynaptic membrane? The membrane which is covering the presynaptic neuron is called as the presynaptic membrane, as the name itself denotes. And then there is a postsynaptic membrane. The membrane which covers the postsynaptic neuron is a postsynaptic membrane, and this helps in formation of synapse. This is what the synapse is. Then we come to another structure which you can see between a presynaptic membrane and a postsynaptic membrane, and that space is called as synaptic cleft. What is synaptic cleft? It is a space between the pre and postsynaptic membrane. Area is around 20 to 50 nanometer wide, and it is filled with extracellular fluid. Neurotransmitters, about which we are going to talk in details in this chapter in the coming classes, are actually diffused through these clefts to reach the postsynaptic membrane. You can see all the structures which we are talking right now in this picture. A presynaptic membrane is there, a synaptic cleft is there, then a neurotransmitter diffuses through this synaptic cleft to reach to the receptors at the postsynaptic membrane. Now these all form synapse. So we were talking about the functional anatomy of synapse. So the presynaptic axon terminal, the postsynaptic membrane, and the synaptic cleft. All of these three form a synapse. Then we talk in detail about presynaptic axon terminal. The terminal which you can see at the top, the axon terminal which you can see at the top, is the presynaptic axon terminal, and this is the terminal which actually receives the information. That the information passes through this synapse. So presynaptic axon terminal consists of a presynaptic membrane, which is the part of this terminal, and then this terminal typically ends in a bulbous enlargement, which is known as terminal button or synaptic knob. So this bulbous enlargement of the terminal end of a presynaptic axon is called as synaptic knob. It is known as synaptic knob. Now this synaptic knob contains two structures. We will again talk about these structures when we see another picture regarding this. We'll see another picture about which I am talking right now. 1 second. Show you the picture. So this presynaptic membrane, which has a bulbous enlargement, and this bulbous enlargement is known as the synaptic knob. This consists of two structures. It consists of the numerous synaptic vesicles. These synaptic vesicles contain, these are the synaptic vesicles. These synaptic vesicles contains neurotransmitters, and then it also consists of a dense tuft. I'm just seeing if the picture is there in this or not. Missing here. I'll share this picture with you in the WhatsApp. I think this picture is missing. I'll share that picture. So this terminal bottom of the synaptic knob consists of numerous seminal vesicles, numerous synaptic vesicles, and it consists of dense tufts. These synaptic vesicles contains neurotransmitters, and there are actually three types of synaptic vesicles: small vesicles with clear core, small vesicles with dense core, large vesicles with dense core. The small vesicles with dense cores usually consist of neurotransmitters like acetylcholine, GABA, and glutamate. The small vesicles with dense cores consist of mainly catecholamines, and large vesicles with dense cores consist of neuropeptide. There is another structure which is present in this synaptic knob is dense tuft. What is dense tuft? Dense tufts are made up of filamentous proteins which have a role in exocytosis of vesicles. These are the dense tufts which are present in the synaptic knob. Then there are large vesicles, small vesicles about which we talked about. The large vesicles are present throughout the presynaptic terminal and are released by exocytosis from all the parts of presynaptic membranes. But the small vesicles lie close to the presynaptic membrane and release to synaptic cleft through active zone by exocytosis. So there is an active zone which is present at the presynaptic knob. These small vesicles are present at that active zone, and thus they are released by the process of exocytosis. This is all happening at the presynaptic axon terminal. Then the region of presynaptic membrane is modified to form what is called as active zone, which contains many proteins, and it also contains calcium channels. When we talk about synaptic transmission in just few minutes, we'll talk about these calcium channels. So these calcium channels helps in release of the vesicles which occurs through active zone. Then there is the postsynaptic membrane, which again forms a functional anatomy of synapse. These contains areas of synaptic transmission which are known as postsynaptic density. So what are these postsynaptic densities? They are clusters of receptors. These are the receptors. These are the clusters of receptors present at the postsynaptic membranes, and they are known as postsynaptic density. Then there is a protein called neurohexine. I am showing the picture. There is a protein called neurohexine. These are the neurohexine proteins. What are these neurohexine proteins? They are actually the binding proteins of synapse, which actually binds the presynaptic membrane with the postsynaptic membrane. This may be the electron microscopic diagram of these neurohexine proteins, which are the membrane proteins which actually binds and helps in formation of synapse, thus providing the structural stability to the synaptic architecture, these neurohexine proteins. Now we talk about steps of synaptic transmission. Here are the steps. First, what is happening at the presynaptic membrane? 5 major steps which are happening at presynaptic membrane. 5 major steps. First, vesicles are concentrated at the active zone of presynaptic axon terminal, and they undergo process called as docking and priming. I'll show you docking and priming diagram. Docking and priming. So what is docking? What is priming? Docking is the process by which the vesicles attach with the membrane. And what is priming? The process in which the vesicles become ready to discharge their content in response to the stimulus. So docking, in which the vesicles attach, the process is called docking. Priming, the process by which the vesicles are ready to discharge the neurotransmitters in response to the stimulus. Action potential comes at the action potential depolarizes the presynaptic membrane. This depolarization leads to opening of voltage gated calcium channels, about which we talked about. They are present in the active zone. So these voltage gated calcium channels open. This results in entry of calcium through the active zone. When calcium concentration increases, there is calcium mediated exocytosis of the vesicles. This occurs and there is discharge of the content into the synaptic cleft. What happens? There is fusion of vesicles, which we talked about, is docking. Then there is opening of calcium channels. Calcium enters into the active zone, and when calcium enters, they help in exocytosis of the vesicles. So this discharge occurs through a hole in the cell membrane, which immediately closes. The discharge of the neurotransmitters from the vesicles happens through a hole in the cell membrane, which then immediately closes. And in this process, the main vesicles remain inside the cell. This is important to know. The vesicles completely don't come out of the presynaptic active zone. They remain inside the cell. Just they discharge their content, and this is called as kiss and run discharge. Kiss and run discharge. Then we talk about the neurotransmitter. The neurotransmitter is released in the cleft. Calcium mediated release of neurotransmitter into the cleft. They diffuses to the postsynaptic membrane, and thus there is quantal release of neurotransmitter, and this is called as Dale's phenomena or Dale's feature phenomena, in which there is a quantal release of neurotransmitter happening and they diffuse towards the postsynaptic membrane. Then we come to role. What is the role of membrane protein? The vesicle membrane is coated with a protein called as clathrin. The vesicle membrane is coated with a protein called as clathrin, which helps in reutilization, and it also helps in neurotransmitter packing. The fusion of the synaptic vesicles with the cell membrane is facilitated by synaptogravin. I'll show you the picture. Show you the picture of the membrane protein called as synaptogravin. Here also you will see, though not very clear, in another picture we'll see. See, these are the two types of protein which are present on the membrane of the vesicles. Synaptogravin, which is present in the vesicular membrane, and syntaxin, which is present in the cell membrane. So this is syntaxin, which is present in the cell membrane, while synaptogravin, which is present in the vesicular membrane. What is the clinical significance, or what is the applied physiology of these two proteins? One which is present in the vesicular membrane, one which is present in the cell membrane. Many neurotoxins inhibit the release of neurotransmitters. Many neurotoxins, neurotoxin means those which are acting on nerves. So many neurotoxins which inhibit the release of neurotransmitters by inhibiting attachment of these two proteins, synaptogravin and syntaxins. And which are these neuroproteins or the neurotoxins? There are two examples for that. These are the two. One is tetanus, another is botulinum toxin. These are the neurotoxins. You must have heard the name of tetanus. So tetanus and botulinum toxins are those which actually, these are the neurotoxins which inhibit the release of neurotransmitter by inhibiting the attachment. You can very well see in the picture, everything is demonstrated here. How they are inhibiting the attachment of synaptobrein and syntaxine with each other, and thus inhibiting the release of neurotransmitters. So thus inhibiting the synaptic mechanisms. So they are the toxins. So this is the clinical significance of me talking to you about these two proteins. Then this was all about the presynaptic membrane mechanism of synaptic transmission. Then in very short, the postsynaptic mechanism. In postsynaptic mechanism, the neurotransmitter attached to the neuroreceptor proteins. I will show you that. See how this is getting attached. These are the receptors. These are the receptors. So these are the receptors with which the neurotransmitters are getting attached, and they bring about conformational changes in the receptors, thus that either open an ion channel or they generate a second messenger. We'll talk about what is second messenger. That will generate change in the ionic permeability of the cell. So what happens when the neurotransmitter attaches to the receptor present on the postsynaptic membrane? They bring about conformational changes, and these conformational changes can do two things. Either they can directly open the ion channel, or they will generate a second messenger which will generate a change in the ionic permeability of the cell. Thus, opening of ion channel will result in movement of ions and change in the membrane potential, and at the level of postsynaptic membrane occur. And this change in the membrane potential is called as synaptic potential, thus creating a signal in postsynaptic neuron. So this was all about the synaptic transmission. We'll talk in details about the synaptic potentials in the next class. I hope you will understand. Thank you so much.